like to thank the organizers for inviting me. It's a great privilege and pleasure to talk to you this morning. I apologize in advance that I can't give this presentation in Spanish, but I hope that my presentation will be clear enough for you to understand the main points. So I've been asked to talk to you this morning about the potential use of neutralizing antibodies for prevention and treatment of HIV infection, and really looking forward to see what we might expect in this space. And this really is built on early work looking at the isolation of neutralizing antibodies from infected individuals. And what we now know is that about a quarter of individuals are shown to induce neutralizing antibodies after about a year or more into infection. And 1% of those actually have potent activity against the majority of viral strains. Cross-reactive antibodies we see a rise over a matter of years, and this is a result of the maturation process of antibody evolution. When we think about antibodies and their antiviral mechanisms, we need to be aware that they come with multiple functions that may have different roles in prevention and in treatment. So not only thinking about neutralization in terms of classical neutralization and binding to viral particles, but also opsonization and phagocytosis, and critically important perhaps in terms of treatment is the ability of antibodies to bind to the surface of infected cells and to trigger through Fc receptor-mediated mechanisms antibody-dependent cellular cytotoxicity. Well, what is becoming increasingly clear is that there are certain sites on the virus that are areas of vulnerability for binding of these potent neutralizing antibodies, particularly the membrane proximal region, the interface between GP120 and GP41, the V1, V2 glycans, and the CD4 binding site. And many antibodies targeting these different regions are now being considered for prevention and for therapy. What is shown in this diagram, looking at the different orientations of some of these neutralizing antibodies, it's becoming increasingly clear that actually the angle of interaction between the antibody and the envelope glycoprotein is critical for them to be able to contact the neutralizing epitopes and penetrate often between the glycan shield. What is exciting in the field is that over the last 9 to 10 years, an increasing number of neutralizing antibodies are being discovered. And what we see is that we're getting antibodies that have stronger and stronger potency and breadth. And what you can see here is that the latest one of these, N6, that was just recently published in Immunity this month, can neutralize 98% of circulating strains in the low nanogram concentration. So this means that antibodies can work at very low concentrations to neutralize the virus. Well, clearly in terms of intervention, the thing that we would like most is to understand how to elicit these antibodies through vaccination. And I'll touch on this a little bit at the end of my presentation. But what people are trying to think about is, having isolated a wide range of neutralizing antibodies, can these now be used as passive infusion to prevent HIV infection or to be utilized in different treatment strategies? So if we think about passive immunization for prophylaxis, we're typically talking about using broadly neutralizing antibodies, often called BNAbs. We know that so far we haven't been able to induce these with vaccine candidates, but these can be manufactured at significant concentrations and be used for passive infusion. So the real question is, what evidence is there that they may work for prevention? And by and large, this is coming out of animal models, but now we're starting to see some clinical trials. So this was first seen with some of the earlier antibodies, particularly B12, was shown in macaque studies that if you treated animals with B12, you could completely block infection from intravaginal challenge. Here you see the B12 treated animals next to B6, which is a non-neutralizing antibody. And what is becoming clear is as you increase the potency of the antibody, you need less antibody to provide sterilizing protection against infection. Obviously, you need to have neutralizing antibodies, shown here, to give that level of protection. Non-neutralizing antibodies don't seem to work at preventing initial infection, but they may have a role at reducing the number of target cells once infection has occurred. And shown in this slide from another study, you can start to see that actually different antibodies have different potency. Here, VRC01 that binds to the CD4 binding site, you can see at a 20 milligram per kilogram concentration, completely blocks infection. It's also potent at 5 milligrams per kilogram in these animals. And then when you go lower, you start to see infection breaking through. This one is an MPR antibody. Again, you can see good levels of protection. PG9, perhaps less potent, less active. Now, potency isn't always the only indication of whether these antibodies work. There may also be other issues like cross-reactivity or short half-life that impact on the potency of the antibody and the ability to provide protection. And then in this more recent paper, looking again at VRC01 and also this other interesting antibody, 3BNC117, you can see that with a single dose of the antibody and then multiple challenges in these animals, you can see that VRC01 can give prolonged protection for a number of weeks against weekly challenge of these animals. And then if you take some of the later antibodies or their modified forms, they can really be quite significant in delaying infection after repeated multiple challenges in these animals. Well, how will they be tested in humans? The first study to look at this in terms of prevention is the AMP study, which stands for Antibody-Mediated Prevention. And this is a study where they're looking at infusing subjects at high risk of HIV infection. The study will be performed by HPTN and the HIV Clinical Trials Consortium in the US. It's a randomized placebo-controlled trial of VRC01. This is the CD4 binding site antibody, and it will enroll subjects both in North and South America and sub-Saharan Africa. The idea is to look at 2 different doses of the antibody to see if there's a dose relationship to protection. It's powered to detect 60% efficacy and to associate plasma levels with protection. Both trials have already started and are well enrolling, and so we should see results from these trials hopefully in the next year or so. Of course, nobody is imagining that a single antibody will be appropriate long-term for prevention or for treatment, and so people are looking at rational design of combinations. And combinations will likely be important both to make sure that we have the right breadth of coverage, but also to increase potency and to reduce the chances for resistance evolving. And this recent paper has been looking at the rational design of antibodies, and depending on the antibodies and the targets that they are going after, if you get the right combinations, you can see that different antibody combinations shift the curve so that you get increased potency and breadth. Now, there will be a cost-benefit trade-off in terms of the more antibodies you include in any prevention strategy, the higher the cost, and so it'll be important to determine whether adding a single antibody up to 4 antibodies in a cocktail is worthwhile in terms of the gains in protection. Well, what about using passive immunization for cure or treatment? Here the interest really is on the ability of antibodies to bind to infected cells, particularly antibodies that may recognize infected cells that either express low levels of antigen or even potentially if there are antibodies that can see latently or very quiescent cells. And this would allow the antibody to trigger, again, Fc-mediated receptor functions and induce ADCC. And the idea is to look at these types of antibodies and see if they can be used in combination with latency reversal agents. And here the concept is that by using these antibodies, we can take the latently infected pool that is not seen by antiretroviral therapy or by the natural host immune response, use the latency reversal agents to wake up the virus, and once it's being expressed to a level that's high enough for antibodies to recognize, this will allow the antibodies to be introduced together with antiretroviral therapy and the host immune response to eliminate the latency pool. So that's fine to think of that in terms of a hypothesis. What is the evidence, again, that this may work? Well, this is currently mainly based on animal experiments. This is another animal experiment from Michel Nussenswag's lab using a humanized mouse. The humanized mouse is infected with HIV and either treated with antibodies or with antiretroviral drugs. They're discontinued and then look to see if there's viral rebound. If you use either of these, it doesn't seem to work. Using antibodies in a combination together with a single latency reversal drug, again, doesn't seem to prevent rebound happening. But interesting, as I'll show in detail, if you use a combination of latency reversal agents, what we see is that that seems to give a level of therapeutic advantage. And this is shown in this slide here. What you can see is that the animals were infected with HIV. You can see the viral load coming up here. They were then treated with multiple doses of this antibody combination, and then the viral load drops. The combination of three latency reversal agents are then used in this period here. And you can see for the animals that are shown in the black lines, the virus doesn't rebound. And in some of the animals shown in the gray lines, you can see there's a rebound after the latency reversal agents. What is promising here is in about 60% of the animals, there was no rebound after this regime. So it would suggest if this type of work could translate to human studies, it might be able to induce long-term remission in HIV therapy. Well, moving to human studies, what's been shown in terms of human studies and passive immunization strategies. This is a study looking at VRC01. Again, it's a CD4 binding site antibody. And this is looking now in chronically infected individuals, not on treatment. And you can see that these individuals, when VRC01 was infused as a single infusion, you can see there's a drop in viral load in the majority of patients. If you look here, you can see two of the subjects had no drop in their viral load, and sequencing of the virus showed that actually the virus that was circulating in those subjects was resistant to this antibody. You can see that some of the individuals had a drop and then returned to their baseline, and two of the individuals seemed to go into long-term remission for that virus following the infusion. A similar study with another CD4 binding site antibody, this time again 3BNC117, shows a similar picture, infusion of the antibody, looking at suppression of viremia. It bounces back in most of the individuals, but some of them, it reduces their set point. Now you have to remember this is a single dose of antibody. It doesn't actually necessarily show what would happen if there were multiple infusions. So it's like giving a single dose of antiretroviral drug and looking at the impact of looking at the impact on the viral load. Well, what is more interesting is the same group from Michel Nussenswag's lab has looked at the ability of this antibody to suppress viral rebound in humans following treatment interruption. In this study, what he did was to take a number of subjects, infuse them with antibody either twice, shown here, 3 times, shown here, and in some individuals they got 4 infusions of the antibody. Now what's modeled on this slide is here in gray is treatment interruption, and these little small ticks on each slide is where the virus would rebound in individuals that didn't get the antibody, and this is modeled on multiple subjects. So this is the normal time to rebound. You can see that there's some delay with some of these patients, but a few of these patients that get multiple rounds of infusion there's a prolonged delay in terms of viral rebound following the antibody treatment. So this suggests that the antibody has some impact on the viral reservoir. If you put all the subjects together, you can see here are the controls and the time to viral rebound versus those that are treated with the antibody. So this is the first evidence that antibodies may have an impact on reservoir and prolong the time from treatment interruption to viral rebound. Now, perhaps the most controversial of the studies that has just very recently come out in October is this study in macaques that has just been published. And this is looking at an antibody now not against HIV, but against alpha-4 beta-7. Alpha-4 beta-7 is the cell surface protein that is expressed on CD4 T cells that home to the gastrointestinal tract. And what this group saw was that if they infused animals with this antibody, it had quite a profound impact on infection. So the model is like this. The animals were infected with the virus. Here it's SIV MAC239. They were infected for 5 weeks. They then received antiviral treatment, and in the control animals shown here, they then had a period of infusion with a non-specific antibody. The antiretroviral drugs were removed, and they looked at both viral load and proviral load, and as you would expect, having only had short-term antiretroviral treatment, the virus comes back in terms of viral load, and there seems to be no impact on the number of infected cells. What was really surprising in this experiment is when they infused the antibodies, the animals with the antibodies against alpha-4 beta-7, what you can see is that a number of animals actually have no viral rebound, and a number of animals actually manage to control viral number of infected cells. So this is very interesting. If you look at the mean between the animals, here are the animals that get the placebo antibody, here are the animals that get alpha-4 beta-7, and you can see the very dramatic impact. Now, whether this translates to humans is something that we need to understand, and quite fortunately, there is already a clinically licensed antibody against alpha-4 beta-7, and so Tony Fauci's group has started a clinical trial to see whether the same type of approach might work in HIV infection. Now, there are a few caveats here. The animals were infected for 5 weeks, and after 5 weeks, antiretroviral treatment was initiated. It's unclear whether this same approach would be effective if the window between infection and treatment was significantly longer. So when we think about what's going on in this space in terms of human trials, there is a number of trials now in place and ongoing with different antibodies, sometimes in acute infection with antiretroviral drugs. Here is the clinical trial looking at the humanized analog of the alpha-4 beta-7 antibody that's just started, and there are a number of groups looking at broadly neutralizing antibodies in combination with latency reversal drugs. So to summarize where we are and what's likely to be happening in the use of monoclonals for prevention and treatment, what we now know in terms of prevention, it is likely to work, but we need to see proof of concept in humans and understand the effective dose. Once we can model the dose relationship in terms of prevention and the amount of antibody, it'll be much faster and easier to understand how to use additional antibodies in combinations to move into that space. This may have a role given the increase in potency of the antibodies, their breadth, and their half-life, and the push in terms of this space is to come up with an approach that would require 3 to 4 subcutaneous injections per year to provide prevention. Now, the competitor in this space obviously would be long-acting antiretroviral drugs, injectable long-acting retroviral drugs, and really what needs to be determined is which approach is most effective and which approach is most cost-effective. In terms of the treatment area, we again have proof of concept that antibodies as drugs can affect the viral load. They can be used in combination with other antiretroviral drugs. What we're waiting to see is any proof of concept that these can affect the cell-associated reservoir for a prolonged period of time before really this type of approach may become mainstream in terms of treatment. Well, using antibodies either as passive infusion or as injection is still going to be quite challenging in terms of reaching subjects in a developing world setting. And so people are also looking at trying to develop long-term solutions to make antibody delivery more efficient. And so in this space, what people are doing is taking the gene sequences for the antibodies and seeing whether they can be delivered as gene therapy. The most advanced of this approach is using the adeno-associated virus as a vector to deliver the antibody-based genes into the muscles and use the muscles themselves to generate the antibodies for prolonged periods of time. This approach has been used with some success to deliver and there are ongoing trials in the UK now trying to model this with one of the broadly neutralizing antibodies, PG9, to see what levels of antibody can be induced and sustained in the systemic compartment. And slightly behind that, others are working at DNA and RNA approaches also to deliver antibody-based genes. And hopefully we'll see some of those entering the clinical space in the medium term. Well, a lot of this type of approach really has been driven by efforts to be able to isolate monoclonal antibodies in infection, and now using the same technology in vaccination, and using that to inform vaccine development. And so individuals can be taken, their B cells can be sorted based on binding to antigen-specific probes into single cells. The heavy and light chains from the antibodies can be amplified. We can use next-generation sequencing to look at the repertoire and ontology of those antibodies and express the antibodies either to look at their binding characteristics and understand how they neutralize, but also to use those antibodies to inform vaccine development. And so looking at the model that we now have in terms of how neutralizing antibodies evolve in HIV-infected patients, what we know is that this takes a period of years and is really very inefficient in that only about 1% of infected individuals go on to generate these broadly neutralizing antibodies. And for those individuals, their virus has already escaped the antibody response. Now, clearly, a vaccine that takes years to induce this type of antibodies is not going to be successful. So what the field is looking at is whether one can take essentially the roadmap that we see in these small number of individuals that make the right type of antibodies and engineer the immunogens so that we can come up with a sequential prime and boosting regime in uninfected individuals and try and induce the same type of antibody responses. And this is really a major challenge for the wider HIV vaccine field in general. What we do know is that by and large, broadly neutralizing antibodies bind to the closed conformation of the envelope glycoprotein, so what we would call the native trimer. But this trimer is not a fixed structure. It's highly mobile. It changes its conformation naturally here. And we know that once it starts to open up, it will elicit a wider range of non-neutralizing antibodies that are immunodominant. And so the field is looking at a range of strategies to design structural immunogens that maintain this closed conformation to preferentially induce neutralizing antibodies. So what we have been looking at in the EVI consortium in terms of our B-cell antigen approach is to take sequences of envelope genes, particularly those that are associated with transmission, to look at designing those, expressing them in vitro, looking at their antigenic profile, their ability to bind neutralizing antibodies in preference to non-neutralizing antibodies, going back, re-engineering them to make them more stable, putting those into animal studies, using the animal studies to feed back again into this iterative design process, and then moving to humans. But even using the humans, pulling out the antibodies that are responding to the immunogens that we put into them so that we can fully understand the repertoire of response that's induced by any vaccine candidate, and use that as a tool to enhance re-engineering of the of the antigens, until we can get a response that gives us the right level of neutralization. Well, people are looking at a range of strategies, and I'm just going to touch on a few of those as examples right now. One strategy is to use a range of stabilizing mutations that stop the virus from opening up, and using those either as native glycoproteins or to engineer them as, to be expressed from viral-like particles as vaccine candidates. Here shows one of the approaches by Rohir Saunders in our consortium, looking at stabilizing these envelope proteins using a SOSIP mutation strategy that allows the conformation to stay in this tight, closed conformation. So you can see here, looking at cryo and electron microscopy, here is the normal type of envelope proteins that people have been working with up till now. This is the new type of envelope protein. You can see it stays in this closed conformation. It's very homogeneous. We start to see autologous antibody responses, and we're using this as a tool to move the immune response towards breadth. Another approach that builds on the same type of structure is to take these immunogens and then use fixation strategies to cross-link these immunogens and make them even more rigid, and see if that gives us additional advantages in terms of neutralization. Now, that alone is not going to be sufficient to develop antibody-based vaccines, and so we're also looking at lineage design approaches where we study HIV-infected patients, particularly those that elicit breadth of neutralization as rapidly as possible, use those envelope sequences to generate new immunogens, use the antibodies to study those immunogens, and look to see if we can come up with a series of immunogens that can train the immune system to move towards a level of breadth that may have preventive use. Well, obviously, to do that type of work, it requires multiple different contributors. So we're lucky to have this new grant that brings in 23 different institutions across Europe. And you'll notice a number of these are based here in Spain, and we're very happy to be part of this collaboration and work with our Spanish partners. So I will finish there. Thank you very much for your attention, and very happy to take some questions.
Thank you very much, Robin. Great presentation. So this presentation is now open for discussion. I can see barely here from here that you have mics on the aisles, on the central aisle, on the lateral aisles. So if any of you want to make a question, please go to the mics and do it. We have Pepe Alcami already here. Go ahead.
Hi, Robin. Thank you for this great talk. I have two questions. One is regarding the treatment as prevention for neutralizing antibodies. Do you think could be a role for these antibodies in mother-to-child transmission at different steps, not only at delivery but also at breastfeeding?
So you're absolutely right. People are already looking at using antibodies for mother-to-child transmission, and actually that may be the most appropriate use of the antibodies, because obviously you need less antibody to prevent mother-to-child transmission. And I suspect that that will be the first area where we see wide-scale use of monoclonal antibodies.
And my second question is about the cost of these treatments, because when you talk about these clinical trials, we wonder if there is a cost, is affordable as a cost. So which is exactly, for example, in this big trial you are collaborating in Africa for prevention, how much is the cost of treating patients with these antibodies?
So that's a very good question. Cost is critically important. So the VRC01 trial is an expensive trial. I don't know exactly what the cost is. The trial is being done for a number of reasons. First of all, it will prove whether neutralizing antibodies on their own can prevent infection. That's important for the field, that's important for vaccine strategies. The second aspect is, hopefully, it will tell us what level of antibody is required to give mucosal protection. If we know what level is required, again, for a vaccine target, we'll know that we need to reach that bar before going into a large-scale efficacy trial. In terms of cost, at the moment, it would be an expensive approach, and most people think about current antibodies that are licensed for use in other infectious diseases, and they are expensive. But cost is related to scale. So if organizations invest in scale-up of the monoclonals, the predicted costs could come down to be really quite low. Now, the time scale for that versus the development of long-acting injectable antiretrovirals, I suspect that long-acting antiretrovirals may be there faster and cheaper and may actually prevent people putting a large amount of money into building big factories to make some of these monoclonals. But I think, it's open and we need to pursue all avenues.
Hello.
We have one question there. Danny?
Thank you for the nice talk. I have a question regarding this article you showed about antibodies that are against this alpha-4 beta-7 molecule in CD4 T cells. I am curious because I wonder whether the authors are proposing an immunological mechanism by which this, by targeting this specific cell subpopulation, they are able to avoid the viral rebound after the treatment interruption. Are these cells maybe dying directly, or they are being activated in some ways?
So that's a critical question, and even the authors of that paper don't fully understand the mechanism. I think what is clear is those T cells are not being killed. Whether they're being activated to be more resistant to the virus is something that we don't understand. And the clear thing is this just an artifact of the animal model, or is this a really strong signal of something that may have potential in humans? I think what is exciting is, rather than that research taking another 10 years to be tested in humans, they have moved straight into a human study. So we will know very shortly whether this mechanism is something really new and extraordinary and worth investing a lot of time in, or whether it's just something to do with the animal model.
Okay, thank you.
So, Robin, we have one question from the table here.
What do you know about the penetration of these antibodies in the CNS, and if this could be an issue if you are going to treat only with antibodies?
Personally, I don't know anything about the penetration into the CNS, and that would be an important consideration.
So, Robin, could you elaborate a little bit more on the issue of resistance to these antibody therapies?
So resistance is clearly going to be a potential issue. And people are looking at using combination of antibodies. We know if you use a single antibody in the same way as if you use a single antiretroviral drug, resistance evolves quickly. Probably it's quicker for antibodies. Now, in the prevention field, resistance is an issue that if you block the infection, then there isn't any viral replication for resistance to evolve. But if the virus gets past it, you may well see resistance, and you may very quickly see that your prophylactic strategy is undermined by resistance evolution. Mm-hmm. In treatment, again, we really don't know enough about whether you use these combinations, whether in the same way that combined antiretroviral therapy can block the virus from evolving, whether these will be sufficient to, again, push the virus to contain resistance evolution.
Okay, thank you. So, thank you, Robin. If there is no other question, you had one more? the last one.
No, my question is about the attempts to induce a specific mucosal protection. So I wonder if there is any IgA broadly neutralizing antibody. So it's.
Yeah, that's another very interesting question. So at the moment, we don't have any naturally isolated, broadly neutralizing IgA antibodies. There are some antibodies that have been engineered from an IgG format to be an IgA format, and particularly Ruth Ruprecht's group in the US has shown that these have been very good for blocking mother-to-child transmission. Whether you need IgA to give you mucosal protection is another big question. I'm not sure that it's essential. If you inject IgG into humans, you see it at mucosal surfaces. And sometimes I think we overestimate the role of IgA. We've looked at IgA in terms of its ability to aggregate virions, neutralize virions. It doesn't seem to be as potent as IgG. So I suspect if you have high enough systemic levels, you will have antibody that is in the tissue right up to the mucosal surfaces and some at the mucosal surfaces, and that should be sufficient. Thank you.
Thank you very much. So we'll be moving to the second presentation.